The title of my talk is the role of mutations in different sodium channels with a focus on the heart. So I would like to address the question whether the other sodium channels are expressed in the heart, and if they are expressed and if they are important, then we should include those channels in genetic testings. Here you can see a phylogenetic tree of the mammalian voltage-gated sodium channels. So there are 10 different isoforms. 1.1 to 3 are the sodium channels in the central nervous system, so they are in the brain. 1.4 is the sodium channel in skeletal muscle. The isoform in the heart is NAV 1.5, or previously called HH1, stands for human heart 1, and the others are expressed in the peripheral nerve system. One important compound in sodium channel research is tetrodotoxin, and it's still very important because we can distinguish between those isoforms because these isoforms 1.1 to 4, 6 and 7 are sensitive for TDX, so they have a high sensitivity. The IC50 is around 10 nanomolar, whereas the cardiac isoform is relatively resistant to TDX with IC50 over 1 micromolar. Tetrodotoxin, as you may know, is accumulates in the buffer fish mainly, so it's an alkaloid produced by some marine microorganisms, so not produced by fugu but by microorganisms, and it accumulates through the food chain, and the lethal oral dose is as low as, let's say, 1 milligram, or less than 5 gram liver if you eat fugu. So this is the title of this paper here: never eat an ugly fish. If you look on the internet, not all buffer fish are that ugly, so it's some are very beautiful. Okay, tetrodotoxin binds from the outside, so this is a sodium channel in the plasma membrane. Tetrodotoxin binds here from the outside. There are several binding sites. The most important binding site is located here in the domain 1 of the sodium channel. It's position 373. As long as there is an aromatic residue, tyrosine, at this position, then the channel is sensitive, so it binds TDX very efficiently. As long as there's a non-aromatic residue, then the channel is relatively resistant to TDX. So we were very interested a couple of years ago in these TDX sensitive isoforms because we actually, in the Friday evening experiment, we discovered these transcripts in the mouse heart. So we didn't want to find them. Actually, I want to clone 1.5 from the mouse, and by a PCR approach, I got 1.4. So I was very surprised, and I thought, okay, there's contamination, whatever, but it turned out that these TDX sensitive isoforms are indeed expressed in the mouse heart. Here you can see the developmental expression pattern. Here are the different isoforms, and here are different developmental stages. So we collected whole hearts and did the RT-PCR analysis. E15 means before birth. There are mainly 2 isoforms, 1.5 and a splice variant of 1.5, but there's almost no or little expression of the others. So these are the TDX sensitive isoforms. So during development, you can see that there's a large expression of 1.3, also 1.4 and 1.2. And in the adult mouse heart, these TDX sensitive isoforms account for almost half of the sodium channel transcripts. So they should be there, and if they are there at these high levels, they should have function, right? So then we analyzed the localization of those sodium channel isoforms by immunofluorescence studies. Here we applied an antibody against the major isoform NAB 1.5. You can see here cardiac myocyte. These are the intercalated discs. So 1.5 is located here, also here, and also the T-tubal system. And a similar localization we obtained for 1.1 and 1.2, so for the neuronal channels. During the last 10 years or 15 years, there are a lot of papers appeared in Medline on these TDX-sensitive sodium channels, and there are many speculations on the function of those sodium channels. For instance, for control of heart rate, for cardiac conduction and action potential shape, or for cardiac contractility. The problem with these studies is that these are all in vitro studies. There are almost no functional studies done with small rodents in most cases, not with higher mammalian species. And so we were interested in the question, are these channels expressed in the human heart, and do they have a function there? If those channels are very important for human cardiac physiology, then we should assume that mutations in those channels, mutations in TDX-sensitive channels, should cause also cardiac diseases, right? That's of course clear. Then second, I thought if those channels are there, then what happens if we intoxicate animals? So there are a lot of such animal studies in the literature, and I checked the literature if there's any effect on the ECG or on the heart or whatever. So are there any effects of tetrodotoxin on the heart or cardiovascular system in animals? And of course, especially in Bangladesh and other countries in Asia, tetrodotoxication is still a matter. Sometimes, so poor people, they sell fugu on the market at a cheap price. So, and then suddenly 20, 30, 40 people may die from tetrodotoxication. There are many case reports also in the literature. So I thought, what happens with those patients, with the heart of the patient? So if the TDX-sensitive channels are there, we should see something. We should at least see effect on heart rate or whatever. And then we analyzed the sodium channel transcript pattern also in the human heart. So, here you can see cardiac action potential with the currents and channels, and there are hundreds of mutations. We had nice talks today, hundreds of mutations here in these potassium channels. And what we know is that if a sodium channel is mutated that affects the cardiac action potential, it is always 1.5. So there's not a single mutation reported in the literature that mutation in 1.4 or 1.2 causes a cardiac problem. There are more than 500 mutations in NAV 1.5, and most of them cause Brugada syndrome, followed by long QT syndrome subtype 3 or cardiac conduction disease. Of course, there are mutations in 1.2 or 1.4. So 1.4 is the channel in the skeletal muscle. If SCN4A or NAV 1.4 is mutated, this causes muscle diseases. If SCN2A is mutated, then this causes epilepsy. So the question is now, is there any effect on the heart in patients having such a mutation? Here you can see the clinical spectrum of SCN2A mutations, and up to now there's no report in the literature that the ECG is altered in those patients, also in muscle diseases. So it is very unlikely that mutations in SCN2A are relevant for cardiac function. There's one point is in epilepsy. Some of patients die because of sudden unexpected death in epilepsy. That is a problem, but many patients with epilepsy do not have SCN2A mutation at all, and they still die. So, and if there is so-called SUDEP or sudden unexpected death in epilepsy, there's always a seizure before. So if the heart is affected, then probably this is due to hypoxia or something like that. So what are now the effects of TDX on the heart or cardiovascular system in animal experiments? Here you can see the number of studies plotted against the publication period. So I did a database search, and you can see that there were already a couple of studies here at the end or beginning of the 19th century, at the beginning of the 20th century. Those studies were done with fugu extracts, so with the liver extracts from the buffer fish. So the problem is that we don't know the exact concentration. TDX was then structurally identified in 1964 and then produced as a powder. So the concentrations in these studies were in the range of 80 to 160 nanomolar, so high enough to block all TDX sensitive channels. So what was the outcome of those studies? Here there are basically two results, and all those studies are very similar. If you intoxicate, for instance, a cat, in this case here it was done 1957, this is a high dose of TDX injected into a cat, then the blood pressure drops and then respiration shortly after stops. So then the cat was ventilated, mechanically ventilated and recovered after a while, so after 1 to 5 hours about. This drop in blood pressure could be due to change in cardiac output or to change in the peripheral resistance. And the authors here showed that this drop in blood pressure is due to vasomotor tone, to reduced vasomotor tone, so due to vasodilation. So the vessels just open. And then if they for instance press on the aorta, abdominal aorta, then blood pressure immediately, instantly is normalized. So the cardiac output was normal in those studies. And here's another study from 1990. Here you can see this is here the drop in, this is the drop in blood pressure here. They intoxicated guinea pigs here. This is the respiratory frequency. So at the final stage, they stopped with respiration. But you can see here when respiration stopped, the heart still continued to beat. This is shown here. This is the ECG here, and this is a recording from an inspiratory neuron. So inspiration stopped here, but the heart still continued to beat. So there was no effect on the heart, even at a very high dose of TDX. This was already published in as early as 1890 by Japanese. At that time they had to publish in German somehow, I don't know why, but very convenient for me to read it. What they concluded with the animal studies is that the mammalian heart is not affected by TDX. So what are now the effects in TDX intoxicated patients? I did a large database search in Medline during this publication period here, and this included more than 500 patients. So you can see the cases in Bangladesh. There are 179, 280 here in Thailand, some in Australia due to the blue-ringed octopus probably. Very few in Japan. They produce fugu in fresh water nowadays, so they don't have the problem. If you go down to the restaurant, it's very safe. Some are in here in Korea. So all the patients were transported to the emergency department and intensive care unit and monitored. So then I thought, okay, if there's some problem with the heart, so it should be reported. But there were very, very few ECG abnormalities in all those patients. So here's one typical example. What happens in TDX intoxication is that sensory neurons are blocked, the diaphragm is blocked, and the muscle is blocked. So mainly sodium channel 1.6, 1.7 is blocked, and 1.4. So 1.1, 2, and 3, they are probably not blocked because the brain is protected by the blood-brain barrier. So the toxin cannot access to those channels. This is a first intoxication case here in Europe. It's maybe due to the changing climate, so the marine microorganisms approached Europe probably, so it happened in Spain. So take care. What was reported here is the general muscle weakness, so 1.4 was blocked. Non-reactive pupils, no tendon, corneal, oculomotor reflexes, so the peripheral nerve was completely blocked. No recordable conduction in sensory motor nerves. They measured conduction, couldn't measure anything. Required intubation, of course, that is essential in that case, mechanical ventilation. So he remained conscious, so he felt everything because the brain still works. and there was a normal heart rate and blood pressure was normal in that case, normal EEG and normal ECG. So then I asked the question, what are the levels of tetrodotoxin in the serum or the blood? Are the levels of toxin high enough to access those channels, and what happens? And what you can see here is the TDX levels in the blood, and here are the 4 degrees in tetrodotoxication. The clinical grading system is based on Fukuda and on the work of Fukuda and Tani from 1941. They established this classification. At a very low level, you have a very little problem, paresthesia, nausea, vomiting, or so not a big problem. And the TDX levels are below in the blood, below the IC50 for the TDX sensitive sodium channels. So at the 3rd grade or 4th grade, they are above this line. So at least at the 4th grade, you should block almost all TDX sensitive sodium channels. And what you see in the patients is apnea, severe hypoxia, hypotension. In very few cases, you see bradycardia and cardiac arrhythmia, and all those patients where you can see some change in the ECG, they have a pre-existing disease like diabetes. So if they are otherwise healthy, you don't see any change in the heart. And at these high levels here of TDX in the blood, already 1.5 is blocked. Of course, the block of 1.5 starts. You may already block, let's say, 10, 20, 30% of the normal cardiac channel, which should affect heart function. So this slide summarizes the effects of tetrodotoxin on the cardiovascular system. And the first, of course, is what TDX is doing, activation of the chemoreceptor trigger zone, causing vomiting. That's very important. Somehow, if TDX is then in the blood, it blocks TDX sensitive sodium channels, 1.4 in the skeletal muscle. In the peripheral nerve system, 1.6, 1.7. This causes muscular incoordination. Then the block of the diaphragm causes respiratory failure, hypoxia. And the block of the peripheral nerve system then causes a reduction of the vasomotor tone, so there's vasodilation. And this causes then the hypotension. Finally, this causes organ failure. And the brain is believed to be protected by the blood-brain barrier, and the heart is protected by its TDX-resistant sodium channel at 1.5. So I would conclude from this that TDX-sensitive sodium channels, if they are on the heart, they are not functional. And first, I come to the last point, to the sodium channel transcript pattern in the human heart. Here you can see the pattern in the mouse. So we isolated whole heart and analyzed by real-time PCR the transcript pattern. This is here the normal cardiac isoform 1.5. This is a splice variant of 1.5. This is 1.3. So the channel in the normally expressed in the central nerve system. This is the skeletal muscle channel. This is 1.2. And then we did this for the rat. So we repeated this many times. So it's not simply contamination or so. In the rat, we have much reduced levels of TDX-sensitive sodium channels. Also, this splice variant is highly expressed. In the dog, we couldn't find this splice variant of NAV 1.5, but large amounts of 1.3, similar as here in mouse. Then we thought, okay, what happens in pig? Pig and human looks very similar, and the only difference here is that the pig expresses another splice variant compared to the human heart, but the levels of TDX-sensitive channels is very, very low. My conclusions, so mutations in TDX-sensitive sodium channels causing cardiac diseases are largely unknown, and there's no correlation between sudden unexpected death and epilepsy and mutations in TDX-sensitive sodium channels. There's no direct effect of a low dose of TDX on heart rhythm, conduction, or cardiac output. And all effects in patients were explained by the block of non-cardiac TDX-sensitive sodium channels. And there are very, very small quantities of TDX-sensitive transcripts in the human heart. So a long talk and a very short conclusion. I think The mouse heart here is probably a photo from Christian. I think you did it in Jena. Here you can see a Langendorf preparation with mouse heart under the microscope. I believe that the mouse is a little bit different, but in the human heart there's only 1.5. That's my conclusion. So thank you very much.
Thank you very much indeed, Dr. Zimmer, for an incredibly interesting talk. I have a question for you, which could be interesting to Dr. Fukada as well. What about SCN10A and its association with Fukada? I say that because our group from Imperial, about a year ago, in collaboration with a group at the Hammersmith who were working on the West London lollipop study. And John Chambers we found that SCN10A is expressed in the human heart from our transplant program. They found in the lollipop study that mutations in SCN10A actually produced delayed conduction in several ways. That was published in Nature Genetics. The story gets a bit more interesting when a group with Philippe Rogel, who is here, looked at whole genome studies and found mutations in this sodium channel, encoded by SCN10A, and I don't know which isoform. You can tell me. SCN10A, were actually associated with a Brugada syndrome, a higher incidence of Brugada syndrome in a population study, several thousand people, when they looked by third generation sequencing, they found this association with Brugada. So what do you think about there is another sodium channel apart from SCN5A, which is associated with Brugada, which in a population study is causing delayed conduction. And in our series of HCM, again, we found that patients with HCM who had, apart from a sarcomeric gene mutation and SCN10A, also had long QD or delayed conduction. What do you think about it?
If this is an important channel in the heart, it's TDX resistant, right? It must be.
I'm not sure whether it's TDX resistant.
It must be TDX resistant. So
I don't know. Is it a TDX resistant? You would know better than me, but I'm just reporting what I know, which is
what is it? I think
incomplete.
it's NAV 1.8, right? So this is TDX resistant. So
is it TDX resistant?
I think so. Yes. All the others are not.
So it doesn't negate what you're saying.
Not really, because we were interested first in the TDX sensitive channels because you can distinguish them from the hot channel.
Why is it in Japan we go to these puff fish restaurants and we don't get intoxicated?
Because they cultivate the fish in fresh water.
They take the liver out.
No, that was done previously, but now they cultivate the fish in fresh water simply.
Fresh water, they don't have TDX.
Seawater, so
we're safe.
Yeah.
Question, huh?
This is like mushroom collectors in Europe. So some people, they of course go for fishing and then they eat fugu. That's it. So that's why there's some people die of fugu, but not from the restaurant. Some Japanese believe that it's very good to eat the liver of fugu.
Puff fish is extremely delicious.
But we
if
have
you
not,
don't die.
Yeah, we have not analyzed SCN10A so far, not by RT-PCR or immunofluorescence in no species, and we would have missed this here. If you apply TDX, so it's same as 1.5.
Yeah.
Dr. Brugada.
Two questions. Two questions that I don't have yet, but I would like to know whether you can help me with that. One is the relation between epilepsy and SAD. It's still something fascinating we hear, that we still do not have more evidence to support that there is a relation, and that this relation relates to the mutation itself, which makes a lot of logic to me. I mean, if you're relating in your brain, when if you relate to brain, you get epilepsy. When you relate to heart, you die. If you have sodium channels in the brain and in the heart, you should have the same final result, electrically speaking. But in one side it kills you, and the other side you just have an epileptic attack. So we should try to understand a little bit more on that, and maybe you can clarify. And the second one is on all these currents that we know. I mean, is there a relation between the amount of current and the importance of this current in the behavior of the heart and the resulting disease of this heart? I mean, for instance, the SCN10A, is probably minority of the sodium channels in the heart relates to this current and can produce redundancy. But the fact that it produces redundancy only in a very specific and limited number, does it mean that you need other things that finally will bring it or will produce a disease? And in SCN5A, because you have a lot of current there, one mutation there produces a lot of effect. I don't know whether this is true or not. Does it make sense? I mean, I'm simply thinking whether the amount of current is related to the amount of damage you can do.
I think it makes sense to cause long QT. I think you need high expression in the ventricles, so you need a large amount of RNA and protein, right? To cause conduction disease, you need a small amount of a specific channel in the conduction system. So if you isolate a heart, you will find small quantities of RNA. But in the conduction system, it may be very important. So the localization is also one factor. So of course the expression in a specific heart region, that is very important. But the problem is that you see which animal model we should use. If we use mice, so what can we find? They probably need these TDX sensitive channels to maintain the heart rate of, if mice are angry, they have, correct me, 800 beats or something like that. So we never can reach such a high heart rate. So they probably need TDX sensitive channels for first upstroke in the science now. So that is which heart we should use to analyze the expression level in the heart. If we, for instance, use heterologous cells, heterologous host cells, so what is that? We have an artificial promoter system, like in short QT syndrome. If there's an increase in the potassium current, this is the proof. It's difficult to say, right? Because what is that? It's just hex cells, or what is it? The promoter is artificial, so what is the mechanism? And the trafficking in the cardiac myocyte could be quite different from the trafficking in the hex cell. But you're right. And your first question was sudden unexpected death in epilepsy. I think mutations in SCN1A and 2A are not a major cause of epilepsy. But many patients die from sudden unexpected death in epilepsy. So many patients without or with a normal sodium channel. And they all. So I'm not a physician, but I heard that they all had seizures before they died. So probably if the heart is affected, it's due to hypoxia or simply. Not simply, but it should be the main cause.